Go ahead and get started. I see a number of students online, and I will, somebody give me a thumb up if you're hearing me okay. I just want to make sure. Good. Okay, thank you. So today we're going to start the next and last section of the course, the endocrine section. So we're going to transition from nervous system to endocrine system, but they're highly related. You'll see some of the same themes. I do want to take this minute before we get into the content of the endocrine section to, give a pep talk a little bit. We've got 2 more weeks in this accelerated mode. So I just want 2 more weeks of working hard for this material is what it takes to succeed. Right now, the weighted course average score is in the middle of the B range. So I guess what I want to say is those of you who are doing well, and there are many of you who are doing well, who are doing, if you're pretty happy with your grade, keep it up, right? 2 more weeks of, the work that you've been doing. We've got, let's see, what do we have? We have 1 problem set due on Friday of this week. Remember, problem sets are open book. I'm happy to answer questions. They can raise your grade if you work on them. If you don't work on them, they don't raise your grade. Then there's a quiz due on Sunday. The quiz will cover the material the anatomy, the names that we cover today and tomorrow and Wednesday. So names of hormones, names of the sources of the hormones, names of the targets of the hormones. So it's not so much anatomy as, where's the thyroid gland? That's not kind of anatomy, but what's the name of the thyroid gland? What are the hormones that the thyroid gland releases? What are the hormones that the thyroid gland responds to? And what are the targets of thyroid hormones? Well, so that will be on the quiz that's due on Sunday, along with leg muscles. So that's your goal for studying for the quiz. Then next week, looking ahead to next week, there's another quiz, the lab practical quiz. And your goal there is to study muscles and nerves and brain structures. We'll spend some time in lab this week reviewing for next week. So there's 2 more quizzes, 1 more problem set, and 1 more exam. That's our work for the next 2 weeks. I'm not saying it's not a light load, but that's what's coming up. So those of you who are doing well, keep doing it. Just keep, whatever you're doing, just power through these next 2 weeks and do the work that you've been doing. Those of you who are falling below your hopes for this course, take some encouragement in the fact that there are these assessments coming up that can help raise your grade if you do the things that you need to do in order to learn and do well on these assessments. So my recommendation is work on it every day. Work on 1 problem on the problem set, and read a couple of pages of the textbook, and write a few summaries of the lecture notes. Work on it for every day, and maybe even do the same thing on successive days in order to help the material form deeper knowledge and understanding in your brain. Don't hesitate to ask Brittany or me questions. But as I said at the very beginning of this course, most of your learning happens outside of this room. So this room is to teach and go over things, but your job is to do this at home. And my recommendation is do it every day. Questions about your future for the next 2 weeks and 2 days? Okay, then let's get started on the endocrine section. So today we're going to talk about types of hormones. We're going to spend some time reviewing the processing of peptide and steroid hormones. We'll before that, we'll talk about sort of the general construction of the endocrine system, and then we'll start talking about hormone receptors. A lot of this is going to be reviewed. GPCRs, you know the story we're going to go to there. Receptor tyrosine kinases will review some of the material you got in self-phys. And we'll talk about some intracellular receptors that we haven't had to introduce in the nervous system. And then we'll start putting it all together into homeostasis. So here is a super oversimplified view of the endocrine system. In the endocrine system, there is a cell, either an endocrine cell or a neurosecretory cell, that releases a hormone into the bloodstream. That is the key difference between that is one of the key differences between the endocrine system and the nervous system. In the nervous system, the signaling cell is interacting directly with the target cell by contact at a synapse. In the endocrine system, the interaction between the sending cell and the receiving cell goes through a blood vessel. So hormones are signaling molecules that are released into a bloodstream or some other vascular system. Endocrine cells are excitable. They fire action potentials. They have voltage-gated sodium channels. They use voltage-gated calcium channels and calcium influx and synaptotagmin and snares and all that stuff that you learned about in nerve cells to trigger the release of hormones. So the release process of hormones from either secretory cells or neurosecretory cells is the same as what you learned in the nervous system. The difference is that the hormone is released into a bloodstream, not into a very constrained synapse. We'll talk about the signals that trigger an endocrine cell to release its hormone. We'll talk about the receptors that hormones bind to. And we'll talk about the effects of hormones in their various target cells. That's where we're going. What is the difference between an endocrine cell and a neurosecretory cell? It's really just an axon. So you'll see some of the cells in the endocrine system have an axon, but then they release their hormone into a bloodstream. And some of the cells in the endocrine system don't even have an axon. They still fire action potentials. They still have voltage-gated calcium channels, sodium channels and calcium channels. They still do all that stuff, but they don't have an axon. You may have noticed so today you handed out, or on Moodle, there is the problem set. Text is due on Friday. The handout has the slides for today, as always, and the key concepts list. And you may have noticed that the key concepts list has some blanks on it, and that was intentional. I'm using the key concepts list as kind of a worksheet, and my recommendation is bring this to class every time, and as we talk about these hormones, fill it in. Use that opportunity to fill in the concept list. I will provide the answers before Friday. So I will provide a version of this that has the answers. But I think filling it in, you writing it in, is part of the process of putting that information in a memory cell in your brain. Okay, so here's the endocrine system. This is what we're going to focus on. Here's the nervous system. This is the autonomic nervous system. Obviously, you've learned some of this concept. The point that I want to make is that the endocrine system and the nervous system both engage in long-distance signaling. They're both signaling from a signal sender to a signal receiver and the receiver can be far away, you know, the blood vessel. So some cell in my brain or my pituitary gland could be sending a signal to my adrenal gland through the bloodstream, meters away. And you know that in the nervous system, it's also long distance. The sending molecule, the sender in the brain, sends a signal out, an axon, and the receiver could be meters away. So they're both long distance. The difference is that in the endocrine system, the signal is broadcast everywhere, right? It goes into the bloodstream, and the blood goes everywhere. It's like, it's like radio, right? I mean, it's sending out a signal into the world, and wherever there's a receiver for that signal, somebody's receiving the signal. But the signal is going everywhere. The nervous system, the cells that are connected from the nervous, you know, from the sympathetic nervous system to the, let's say, to the heart, that's not quite a meter, but it's a long distance. They're connected by the axon. They are physically connected by a structure, the axon. So the signal is not going everywhere. It's going to its receiver. It's like a landline telephone, right? I mean, these analogies of radio and telephone are starting to break down these days, but you know what I'm getting at. There's a direct connection between the sender on one end of the phone and the receiver at the other end of the wire receiving the signal. Think of landline telephone. So in one case, they're both long distance, but in one case the signal's going everywhere and then being received by specific receptor cells that are specialized for receiving the signal. Because the signal's going everywhere, but it's not telling every cell in your body to do the same thing. It's telling the receiver cell to do something. In the nervous system, the connections are made during development. When your body is being developed, those connections are being made, and those connections are specific and direct. We're not going to talk about the exocrine system. This is secreting things outside of your body. So we talk about exocrine functions in AMP2. We don't talk about sweat, but we do talk about the secretion of enzymes that are necessary for digestion. That's exocrine function. We're not going to do that now. I just want to remind you, some of you, that that exists. There is also a variant of the endocrine system called the autocrine-paracrine signaling, which is not long distance. So the main difference between endocrine and autocrine-paracrine is that this is local. The sending cell is either the same cell or it's nearby cells. It does not rely on a bloodstream. It just relies on simple diffusion to get the signal from its sending location to its receiver. Autocrine just means the cell that secretes it is also responding to it. And paracrine means that the cell that sends it is within diffusion distance of the cell that receives it. And we'll see autocrine and paracrine signaling when we get to some reproductive anatomy, physiology. Okay, so some key similarities, some key differences, some key similarities, some key differences. Those are things that I'm going to ask you to write about on the problem set. All right, let's actually do a clicker. So I know we haven't done a clicker in a long time in this room, but let's, and some of you probably, oh, we'll do it. If you have your clicker, get out your clicker. If you don't, we'll muddle through. So take a minute, let's take a minute to do this clicker. And show me your answers. All right, those of you with clickers, show me your answers. I think we'll go through them one at a time, so if you don't have your clicker, just be prepared to say yes or no when we get to A. Is A true or not? Okay, so I think I have, let's go through these. So, is A a true statement? Yeah, that's pretty much the definition of the endocrine system. It requires a bloodstream or a vascular system. So A is definitely true. Is B a true statement? Yes, that's true. Those are the definitions of those systems. So A and B are basically a review of the definitions. C, is C true? C is a true statement. Why did you know that that was true? Because in the endocrine system, the hormone is released into the bloodstream. And in a human, we've got 5 liters of blood. So the cells, which are small, are releasing the hormone into this huge volume. And so the concentration of hormone, when it reaches its target, is very low, picomolar sometimes. Well, in order for the target to respond to very low concentrations of hormone, it has to have very high affinity for the hormone. So that's what makes C a true statement. In the nervous system, the neurotransmitter is released into the synapse. The synapse has a very small volume. The concentration of neurotransmitter in the synapse is not very low, millimolar, millions of times higher than a hormone concentration in the bloodstream. The volume is very constrained, but the concentration is not low. So neurotransmitter receptors did not evolve to be with such high affinity. They evolved to be expressed in the right place, but they did not evolve the necessity for binding nanoscopic concentrations of hormone. So C is a true statement. D, you could argue either way, right? I mean, you could argue that these nerve cells with an axon and dendritic arborization and arborization of the synaptic terminals. Remember, we've got nerve cells, they've got the complicated dendrite, and then a cell body, and then an axon, and a complicated dendritic branching. That's morphology, and you could say that that's more complicated than that. But you could also say, well, a cell is a cell is a cell. This morphology, we're not seeing the nucleus and the endoplasmic reticulum and the mitochondria and this intracellular, cytoskeleton. We're not seeing all that, and all of that's here and here. So that's why D is a discussion question. You could argue it both ways. I actually think that D is true, but maybe that's just my bias as a neurophysiologist. Okay, so here's a table of where we are going. So I don't like to talk about tables, but this is a reference source. So we're going to talk about the adrenal and the ovary when we get to reproductive endocrinology. We will talk a little bit about the pancreas, but frankly, this organ is mostly discussed in AMP2. So we'll mention this a little bit with regard to some hormones, but I've split the pancreas into the AMP2 part and the AMP1 part. We're in AMP1. We'll talk a little bit about this. We will not talk about the exocrine pancreas because that's AMP2. We'll talk about the pituitary big time, because the pituitary is sort of a master controller of the endocrine system. So we're going to talk about pituitary a lot. Parathyroid a little bit, because it's related to the control of calcium, which I think is important. Testes, the male reproductive system, we'll talk about that. And thyroid. We will talk about all of these things. This is your summary reference table. we will talk a little bit about the organs in your body that you don't think of as being endocrine. So for example, you don't think of your kidney as being an endocrine organ. You think of it as being an excretory organ, controlling blood volume, controlling blood salts, controlling, helping to control blood pressure, etc. But it's an endocrine organ because it releases a hormone called renin. But this is covered in AMP2. So this endocrinology is covered in AMP2. This endocrinology of your digestive system, it turns out, is controlled by the endocrine system and the nervous system. These hormones of the gastrointestinal tract, the digestive system, are covered in AMP2. So we're not going to cover the endocrinology of the digestive system because that's AMP2. We're not going to cover the endocrinology of the heart because it's a little bit aside. We're not going to cover the endocrinology of the kidney because we cover that in AMP2. We'll mention the endocrine function of the placenta. The placenta's main job is to provide nutrition for the growing fetus, but it's also an endocrine organ. We'll mention that in the very last class, when we talk about pregnancy. We're not going to talk about eicosanoids. We're just not going to talk about it. And we're not going to talk about another endocrine organ called the pineal gland, which is in the middle of your brain, and it releases a hormone, and we're just not, that's, the concepts, we're going to stay focused on the main concepts here and not talk about some of these other details. So that's where we're going. That's the roadmap of where we're going. What I would like to do now is transition to talking about the structures of hormones. What are hormones? Well, they fall into three categories. Amines, which you have already heard of. Some of them are the catecholamines that are neurotransmitters. So some neurotransmitters, like dopamine, are a neurotransmitter when they're released by neurons, but are a hormone when they are released by neurosecretory cells. So dopamine is both a neurotransmitter and a hormone. Norepinephrine and epinephrine. The other catecholamines are also both neurotransmitters and hormones. And you know about these, so I'm going to just review those quickly. It turns out that the thyroid hormones are also derivatives of tyrosine, but they're not catecholamines. They're structurally very different, but they are amines. So the thyroid hormones fall into the category of amine hormones. We'll talk more about those when we get to the thyroid. A lot of the hormones in your body are small proteins, polypeptides. Some of them are very short, just a few amino acids, and some of them are actual proteins with a couple dozen, maybe 100 amino acids. They're proteins. So they're synthesized by all the protein machinery that you learned about in cell phys. So we're going to review that. But they're secreted proteins. So not only are they synthesized by all that machinery that you learned about in cell phys, but they're secreted. They're synthesized into vesicles. So there's that co-translational translocation story that you learned about in cell phys, and I'm going to reinforce that. So lots of your hormones are small to medium-sized proteins. And some of your hormones are steroids. And we'll talk about the structure of steroids in a minute. And it's not just the sex hormones. Cortisol, one of the most important hormones in your body because it's required to survive, is a steroid hormone. So let's talk about the amines for a minute. So now let's talk about the amines. Here is the story. You've seen this before when we talked about the synthesis of the catecholamines. There's the catechol, there's the amine. Catechol, amine. Catecholamine. All are derivatives of tyrosine. I do not expect you to learn to memorize the order. I don't expect you to memorize the names of the enzymes necessary to synthesize the amines. Don't do that. But there's a pathway from tyrosine to dopamine that requires two specific enzymes to make it. Enzymes catalyze the reactions that make dopamine. And then another enzyme catalyzes the reaction to make norepinephrine from dopamine. The point is, you've seen this before. I do expect you to be able to recognize the catecholamine group, recognize when something is a catecholamine. I do not expect you to be able to recognize dopamine as different from epinephrine. That's not something I expect you to do. And I don't expect you to know the names of the specific enzymes. But if I gave you a problem and I told you that the rate limiting step for the formation of all the catecholamines depended on the enzyme tyrosine hydroxylase, I might be able to ask you a question to work through a problem related to a defect in tyrosine hydroxylase, right? That if you had a defect in tyrosine hydroxylase, all of these hormones, dopamine, norepinephrine, and epinephrine, the production would be impaired. Because they're all downstream from the first step. Right? That's the sort of thing. So I could expect you to work through a problem if I give you the right information. The other amines are the thyroid hormones. We have two thyroid hormones. T4. It's called T4 because it has 4 iodides. So this is an iodinated derivative of tyrosine. You can see the tyrosine ring group. There's the amine of tyrosine. There's the tyrosine ring group. These hormones there's an enzyme that moves one ring from one tyrosine onto another tyrosine to make this 2-ring structure. So there are enzymes necessary to synthesize thyroid hormones from tyrosine. And then there are other enzymes necessary to put iodide on the rings. You may know that iodide is an essential nutrient. Your body doesn't make iodide. You get it from your diet. If you don't eat trace amounts of iodide, you can't make thyroid hormone. The iodination is required. And so in parts of the world where there is no iodide in the food sources, people develop thyroid hormone problems. We'll talk about that when we get to the thyroid, because of a dietary deficiency. There are parts of this country that have low iodide diets, and that's why the salt companies, if you look carefully at your canister of salt, some of them say iodized. That means they're putting trace amounts of sodium iodide into the salt. There's sodium iodide mixed in with the sodium chloride of the table salt. And that gives, you can't taste it, it's at an extremely low concentration. That means when you put salt on your food, you are giving your body a little bit of iodine. Most of us get enough iodine from our diets. A decent mixed diet with, protein, especially fish, fresh vegetables, that diet provides enough iodine. But in order to make sure everybody gets enough iodine, the FDA has asked the salt companies to prepare a product that has iodine in the salt. And you can buy non-iodized or iodized. And if you think you might be, I mean, I'm not going to get on my soapbox about eating iodide, but it provides, the iodized version provides enough iodine for everybody, regardless of your diet. Anyway, bottom line, making this two-ring structure from tyrosine and iodinating it are required to make the thyroid hormones. There is another hormone that we're not going to talk about very much because it's from the pineal. It's called melatonin. You've heard of it. It's related to sleep and circadian rhythms. It is an amine, but it is not derived from tyrosine because it's derived from tryptophan. So, having shown you this slide, I could ask you to know that melatonin is an amine, but is not a derivative of tyrosine. All the others are derivatives of tyrosine, as well as being amines. Okay, that's a fact that I could quiz you on. Okay, that's it for the amine hormones. Now let's talk about the peptide hormones, protein. Well, now I am going to rely on your memory, your knowledge from cell phys. I'm going to rely on it, and I'm going to build and reinforce it. So you know that proteins are synthesized from mRNA and tRNA and amino acids to make a protein. Right? So the green blob here is the ribosome. The yellow strand here is the mRNA that codes for the protein. We don't see the tRNA that is bringing, oh there it is, tRNA is the transfer RNA is bringing the amino acid, the amino acyl, so that the ribosome can attach it to a protein, which this is the black and the red line are the emerging, synthesizing protein. So we have a ribosome mRNA coding for it, and the protein coming out. That should look familiar from cell phys. All of the hormones that we're talking about now are secreted, right? So they're all secretory proteins. So they all have a signal sequence at the N-terminus. They all have the signal sequence that tells the rest of the cell, and we're going to get to the rest of the cell in a minute. I am a protein that needs to be synthesized, but it doesn't belong in the cytoplasm. It belongs in a secretory vesicle so that it can be exocytosed. That's what the signal sequence at the N-terminus tells the cell. How does it do that? Well, there's a protein called signal recognition protein that binds to the signal sequence at the N-terminus. Remember, the N-terminus of a protein is synthesized first. So after the first 20 amino acids are synthesized, the signal is there, and the signal recognition particle recognizes it and binds to it. And the combination of the signal recognition particle being bound and the ribosome being sort of stalled gets the ribosome to bind to this translocation machinery in the endoplasmic reticulum membrane. So this is cytosol, this is the ER membrane, and this is the lumen, the inside of the endoplasmic reticulum. So we form this complex, and this complex docks onto this translocation machinery in the endoplasmic reticulum membrane. And this docking releases the signal recognition protein and allows translation to continue because the signal recognition protein stalled translation. So when the signal recognition protein is unbound, because the ribosome has docked to the translocation machinery, then synthesis can continue, but now we get that transmembrane translocation. The protein as it's being synthesized is synthesized through the translocation machinery into the lumen of the endoplasmic reticulum. So we directly synthesize these secreted proteins from the ribosome, not into the cytosol, but into the lumen of the ER. And it depends on the signal recognition protein. Well, it depends on the signal sequence. It depends on the signal recognition protein. It depends on GTP. You didn't learn about that in cell phys, but it's true, and I'm not going to get into the details, but there is energy involved, hydrolysis of GTP. I'm not going to test you on that, but it shouldn't come as any surprise to you that there's more to it than what you learned about in cell phys, and in fact, there's more to it than I'm teaching right now. But there is hydrolysis of GTP associated with getting the signal recognition particle off. There's certainly hydrolysis of GTP associated with protein translation. But the point is, let me get to the main point, signal recognition, transmembrane synthesis of the protein. And the protein ends up in the lumen of the endoplasmic reticulum, where it will be stored, processed, and get ready for secretion. So let's talk about the processing. The first step of processing is cutting off the signal sequence. The signal sequence is cut, and so the enzyme, the hormone, ends up in the lumen here without the signal. Now there is some jargon that I need to introduce specifically related to the endocrine system. The protein that is synthesized we call the pre-pro hormone. So let's just unpack this word. Hormone is the thing that we're trying to make. A pro-hormone is a precursor to the hormone. And pre-pro hormone is a precursor to the precursor. So we're going to have at least, in most cases, in many cases, we're going to have two steps of cutting. We're going to cut the pre, and then we're going to cut the pro to make the hormone. Cutting the signal sequence is cutting off the pre part. So the hormone that is, the thing that is secreted, the peptide that is synthesized, is the pre-pro hormone. And then cutting off the signal sequence turns that pre-pro hormone into a precursor of the hormone. And that is then subject to additional cutting and processing. It's just, I want you to know that the name of the protein, generic protein that we are synthesizing by the ribosome, is called the pre-pro hormone. So if we talk about, there is going to be, let's just think about a peptide hormone, insulin. There is going to be pre-pro insulin, then there is going to be pro-insulin when the signal sequence is cut off, and then there's going to be insulin after the processing is complete. Pre-pro-insulin, pro-insulin, and then insulin. And that will be true for most of the peptide hormones. So what happens in the endoplasmic reticulum? Well, again, this is getting back to cell phys, reinforcing things. We've got folding of the pre-pro hormone. Well, let's just say we've cut off the signal. So we've got folding of the pro hormone. maybe some formation of disulfide bonds, depends on the hormone. Probably some additional cutting to cut the pro-insulin into insulin. So insulin, as the case, requires both folding and disulfide bond formation. Just turns out insulin has 2 disulfide bonds, and additional cutting to make the final insulin hormone. And then the hormone is concentrated, the hormone is processed through the Golgi stack, concentrated into vesicles, trafficked to their secretion sites, and then there is a stimulated secretion. Almost all the hormones we're going to talk about are secreted when they are stimulated, and the stimulation will be action potential, depolarization, voltage-gated calcium channels, calcium influx, synaptotagmin, snares, fusion, exocytosis. That story that should sound really familiar. So questions about this general process of processing peptide hormone. Let's talk about a specific case of processing, because this is an important general case. So this is a pro hormone. What does that mean? It means that there was a signal sequence up here that was already cut off, because the pre-pro hormone was with the signal sequence. That's gone. So we have a pro hormone. And this is just a linear map of the pro hormone. N terminus, C terminus. Right? At the N terminus, there used to be a signal sequence, but it's gone. This is a pro hormone. This is the pro hormone for a really important hormone called ACTH. ACTH is important because it controls cortisol, and cortisol is important because your body needs it to survive. So this is an important precursor to ACTH, and ACTH is important because of cortisol. So you can see that if we cut this pro hormone here and here, we generate ACTH. But if we cut ACTH in half or so, we destroy ACTH and we generate a different hormone called MSH. So it depends on what cutting happens. And the cell, it's sort of a semi-random process. There's some control, but there's some not quite controlled. So when the enzymes in the lumen of the endoplasmic reticulum are working on this pro hormone, which enzymes work fastest and which enzymes work first is what controls what hormones are produced. So, for example, the enzyme that cuts here and here is probably pretty active because it's making the essential hormone ACTH. But before this folds, the enzyme that cuts here could destroy ACTH and make a different hormone. It's semi-random. The point is, the endoplasmic reticulum that is receiving POMC, the pro hormone, is making multiple final hormones depending on the cutting. So it can make ACTH. If it cuts here and here, it makes enkephalin. This is enkephalin. This is an endogenous opioid. You've heard about enkephalins, runner's high, or related to opioid addiction. So the same precursor for ACTH is the precursor for enkephalin. And it's also the precursor for endorphin, another opioid hormone. And it's the precursor for MSH. And it's the precursor for some other hormones, LPH. The point is the prohormone is the precursor for multiple final products. And I'm going to ask you to work with that in a problem set. I will not ask you to I do not memorize the map of POMC. Do not memorize the map of POMC. I do want you to know that the precursor for ACTH is the precursor for enkephalin and endorphin and other hormones like MSH, and the name of it is POMC. Pro-opio melano cortin. Cortin that comes from cortisol. So there's the cortin part. There's the melanocyte part. Melano part. And there's the opio part. So alternative cleavage makes all of these important proteins. What do melanocytes do? Let me just ask that question. So we're going to talk about the adrenal, put that aside. We're not going to talk about opioids too much, put that aside. We're not going to talk about melanocytes too much, but just to be clear, what do melanocytes do? They're important for skin pigmentation. So freckles, dark skin, tanning are the activation of melanocytes in the skin. All right, let's talk about steroid hormones. So we're done with amines, we're done with peptides. These are the steroid hormones. You've heard of them: testosterone, estrogen, progesterone, the female reproductive hormones. We're going to talk more about aldosterone, but mostly we talk about this in AMP2. We're going to talk about cortisol, because cortisol is a steroid hormone that's required for life. They all are derivatives of cholesterol. So I do not expect you to memorize the structure of these hormones, but I do expect you to recognize this four-ring structure of cholesterol that is conserved in all of these steroid hormones. They all have the four-ring structure from cholesterol. So you may have heard that cholesterol is bad, and yeah, too much cholesterol is bad, but you need some cholesterol, because if you don't have some cholesterol, you don't have the precursor for cortisol, which is required. So cholesterol has some very important good sides to it, not just membranes, but as a precursor, not just structure of membranes, but as a precursor to the steroid hormones. So as you can probably predict, there are enzymes that control the synthesis of the steroid hormones from their precursors. And here's a map of the enzymes and the precursors. So what do I want you to know from this? I want you to know that they all come, the most upstream the source is cholesterol. I want you to know that they all come from cholesterol. So here is aldosterone, through multiple steps to make aldosterone. Cortisol, multiple steps to make cortisol. Multiple steps to make testosterone, multiple steps to make estrogens. These codes in the italics are abbreviations for the enzymes necessary to catalyze that step. So you can see, I am not going to, do not memorize the order, do not memorize the map, do not memorize the name of the enzymes. Don't do that. I will ask you to work with this though, because, for example, if you had a genetic defect in this enzyme, the P450 17-alpha hydroxylase, you can see that production of cortisol and testosterone and estrogens is all going to be impaired because all of this requires this enzyme to get there. So a key concept is, in terms of answering problem questions, is if there is a defect here, there's going to be no problem making aldosterone. In fact, there might be excess aldosterone because here, when you get to progesterone, alternate processing is impaired. All of the progesterone, if this is impaired, all of the progesterone is going to end up as aldosterone instead of some of it ending up as cortisol and some of it ending up as testosterone and estradiol. So the processing could lead to defects downstream, that is to say deficiencies downstream from the defect, and excesses upstream from the defect. That's the concept that I want you to be able to use in solving a problem with this map in your hand. And so some of the problems are related to this map. I could give you this map on an exam and ask you to work with it. Here's something interesting. The estrogens are downstream from the androgens. So testosterone and androstenedione, the enzymes associated, the hormones associated with maleness, they're called the androgens. The androgens are the precursors for the estrogens. There is an enzyme, the P450 aromatase. Again, do not memorize the name, but it's interesting that the estrogens require this enzyme to convert the androgens. And you can imagine that there are some, physiological effects. If a woman develops a problem with the P450 aromatase, excess androgens in that woman, and low estrogens in that woman. And that's going to have some physiological effects, as you can imagine. Yeah.
During the mental period of work, I guess also substances of choose, like some external effects goes to alter the concentration of that enzyme. Would the individuals develop differently as a result of different
Yeah, absolutely. So there are people who are genetically female. That is to say, they have genetically female means they have two X chromosomes, but they have a defect in P450 aromatase. So their gonads do not develop typically during development. Those are girls that are sometimes identified at birth as boys because of the way that they have what is called ambiguous genitalia. And so there have been cases where a genetic girl, XX chromosome, was identified as a boy because of the ambiguity of their genitalia and because of the excess, the ambiguity of the genitalia came from the development of male, like reproductive organs, rather than female. And there are cases of not only ambiguous genitalia, but an ambiguous sexual orientation as a result of a alteration in their enzymology. Did that answer your question?
Yeah, I think so.
So let's stop. Let's stop with a clicker. I'll let you do this during the break. We'll take a 10-minute break, and then we'll start with the next topic, which is we're going to get into the receptors. So let's take a 8-minute break and then do receptors.